seminar of the fall semester, so I'm going to do just a little introduction. I'm Marcy Brandenburg. I'm the bioinformationist with the Top Mineral Sciences Library, DCMB, and the Bioinformatics Core, and I do the programming for the seminar series, so if at any point in time you got any questions or you want to present, I would be the person to contact. So there are some new faces, so if you've not been here before, this is a weekly seminar series. We basically has a different focus than a lot of other seminars based on its name. We focus on tools and technology and methodologies. Really, sometimes they, are just tools or technologies that are being used in a way that may be of interest. Sometimes they're new tools and technologies. Other times they're actually still in development, and that's perfectly fine. Maybe you just want some feedback on that tool. So there is a sign-in sheet. I'm going to pass that around. Please do sign in. It just justifies the pizza, and we want to continue offering pizza. So with that, I'm going to introduce our speaker, Michael Brooks, who is a postdoc in hematology and oncology in Max Fish's lab.
So yeah, is the microphone sound good? Okay. So as Marcy said, my name is Michael Brooks. I'm in the Wisha lab. I do a lot of breast cancer sequencing. My background is in both wet lab and dry lab stuff. I did my PhD at WashU, and the first half of my PhD was very computational, doing sequencing technology stuff almost as it came out after Barbara Wold's RNA-seq paper, and I was actually working on methods of improving the quantification of sequencing at that point, and then later moved into the world of cancer, and then came here to the University of Michigan to join Max's lab and do a lot of cancer stuff. So today will be pretty biologically focused because that's how I tend to do a lot of stuff, is on the biology side, but any computational questions that I do miss, feel free to ask them as I'm going along. So as I mentioned in the title, today we're going to look at breast cancer stem cells and using Drop-Seq to look at the heterogeneity in those populations. So what are cancer stem cells? So basically, in 1996, John Dick basically showed, and was later shown in solid tumors, that there were a small population of tumor cells that had increased tumorigenicity relative to the rest of the population. They were also shown to have unlimited self-renewal, and they could recapitulate the original heterogeneity of the tumor when you took them out. So, we called them cancer stem cells. The nice idea behind them is that maybe they are what are really driving tumor growth, and being able to target those cells and eliminate them might go a long way in being able to treat cancer. So in a conventional therapy model, which is designed to treat the fastest cycling cells of a particular tumor, most of those type of modalities are not designed to target cancer stem cells, and the cancer stem cells remain behind and are thought to really drive tumor relapse. So the idea is that if you can find specific therapies to eliminate those, particularly in combination, we think that this model in itself would be a little outdated. Than if you targeted just the cancer stem cells, it still wouldn't be quite enough. But being able to target both the regular cells and the tumor stem cells would go a long way to being able to treat cancer. Now, in the Wisha lab, we were Max, and later, in collaboration with a lot of other people, found these cancer stem cell markers in solid tumors. The first set of markers that was used was CD44 plus 24 minus. And later, we also used ALDH. And I only mention that because we'll talk about that a little bit later for some of my data analysis. Now, lately, this idea of cancer stem cells is a little bit of a controversial field in cancer. It comes down to somewhat semantics, whether you believe they're truly stem cells or whether there's just a population of cells that are more tumorigenic than the others. But recently, a lot of work has kind of backed this up, and this was a really nice paper that probably most everyone in here is familiar with. That came out just a little while ago, where they looked through the TCAG data and basically showed that there was a very strong Stemness signature that was associated with dedifferentiation and this ability to have increased tumorigenicity. And so kind of backed up that, the cancer stem cell hypothesis, even on large data sets, is probably correct. And so this fits well with part of our hypothesis in the Wisha lab, trying to look at kind of this normal hierarchy and what are we trying to really see with cancer stem cells. So one of our ideas is that, this is the Waddington's landscape for anyone that's not familiar with it, and it's a very old figure, and it basically was this idea that as cells develop in developmental time, they start to flow down basically the path of least resistance. But there's blocks between going either back up the hill or across divides, and so that's how cells going from a stem cell to a progenitor cell to a differentiated cell, they kind of follow these various paths, and why, once you're in a final kind of normal human developmental stage, you have a lot of these very specialized cell types, and that's how you get there. So basically, as you go back up the hill, you have increased stemness, increased plasticity, and ability to go down these different channels. And so if you take any particular tissue, for example, breast, it can differentiate down a couple different pathways, luminal and basal cells. But the important part is, and what we think is happening, is that oncogenic transformation increases the stemness of the particular cell that gets the oncogenic event and leads to somewhere in the middle of a breast cancer stem cell. And we know that they don't go fully back to complete pluripotency because breast tumors look different than colon tumors, look different than brain tumors, etc. And there are a very rare type of tumors that kind of can do everything called teratomas that are more from all the way up to embryonic stem cells. So this is important. So we can, what we're trying to hope to eventually be able to see is signatures of this increased stemness, find the properties of these cells, look at how heterogeneous they are they able to transform down these different pathways that lead to tumor cells and all sorts of other cells that facilitate tumor growth and metastases, and be able to then know the difference between this cell versus the rest of your normal breast stem cells or other colon cells and things like that. So particularly in the Wisha lab, when I joined the lab, I was really interested in this heterogeneity, and this was actually, in 2014, so certainly single-cell sequencing was around, but Drop-Seq hadn't come out, and this idea of being able to do really high throughput single-cell sequencing wasn't around, but I was still really interested in heterogeneity and understanding the whole gamut of cells that are present in the tumor. Now, in tumors, there's two different types of heterogeneity for the most part. There's genetic heterogeneity, so essentially every tumor develops genetic mutations over its life, and those kind of build and compound on each other. And at least in the Wisha lab, we do not focus on this too much. What we're really interested in is epigenetic heterogeneity. So even given the same set of genetic mutations, we know those cells can interconvert between different stem cell types and produce different types of mesenchymal and epithelial cells. So in any given tumor, you might have a couple different types of stem cells, and these various stem cells, again, as I mentioned, on the the mesenchymal side here were marked by CD44 plus, CD24 minus. On the more epithelial side, marked by being ALDH plus. And we know that these can interconvert, and that's probably mediated by the tumor microenvironment. But each of these stem cells themselves can probably differentiate down the mesenchymal and epithelial lineages respectively. So again, any given tumor might have all of these tumor cell types, or maybe a very specific subset of them. And so what we're really interested in is using that information to be able to eventually characterize any particular tumor as a subset of these various cells. And so we think in a very broad sense, there's all these different molecular subtypes of breast cancer. So you have, most people have probably heard of like triple negative breast cancer. So that encompasses both these top two, but there's an even kind of subdivided part of triple negatives called the Claudin lows that are very aggressive and highly mesenchymal. Basal is kind of the more common triple negative. It's still mostly epithelial cells, but with a high percentage of mesenchymal cells. And this goes all the way down to like a luminal A tumor that would be almost entirely bulk epithelial cells. And so our goal is to eventually be able to take a tumor and essentially kind of look and prove this hypothesis. Look at the various different populations that are present in any given tumor and be able to kind of bend them into their various cell types. And also look at a bunch of different tumors and understand that. And the last part of this kind of hypothesis is what we suspect is that a mesenchymal cancer stem cell from one tumor might be very similar to a mesenchymal stem cell from a different type of tumor. They might be in different proportions. Some tumors might not have them, but we think that they would use the same pathways to drive their functions in the tumor. And we hope to then be able to use that to kind of look at targeted therapies for various different tumor types and come up with stuff that might be able to target as many tumor types as possible.
Can I ask a question?
Absolutely.
Is there any reason as to have like a discrete number of bins in each of these as opposed to a continuous spectrum of more continuous spectrum?
No, I mean, we think that probably there aren't, an infinite number of cell types in any of these, but absolutely, like, if at the very least the bins are so overlapping that they would probably, at a rough estimate, look pretty continuous. But if you sampled enough, you'd probably start seeing bins because at least in normal biology, there's very defined bins. Cells go, there's a very short transition period and they go to a different cell type and they go to a different cell type. And we think that in general, cancer kind of certainly will kind of make those boundaries muddier, but they probably don't completely disappear in cells, kind of have different cell types, even in tumors, that we think of as kind of, in that sense, very homogeneous. So we also know that a lot of these cells, are not sitting in isolation. There's feedback loops between the various cell types that contribute to these different models. And while I won't talk about it a lot today, the second half of my project is both describing what the cell types are present and looking at the population dynamics, so understanding how the cells change over time. So, just to understand what our ultimate goal is understanding that when you could draw a network between all of these, of course, and kind of use, Markov chains and to understand all these different things. Though while, of course, some of these would be zeros, what we're really interested in is for any given cell population, defining all the transition probabilities and all of the genes for this population. But essentially now we know, of course we knew this before, but now we can actually look at these instead of as like single numbers, using single cells to get populations for all these, and really understand what is happening at each of these nodes and what would happen when you eliminate one of these populations. Because as going back, we know that even, for example, when you might eliminate all of the non-cancer stem cells with a conventional therapy, the cancer stem cells will recapitulate the rest of the tumor. And if you even remove the cancer stem cells, you can get dedifferentiation, and those cells can come back and make cancer stem cells. So you have to really get a full picture of the entire thing to be able to know what might be the key linchpin tumor type that you could remove to be most effective. So I certainly won't go too much into the background of Drop-Seq, but there might be a few people here that aren't familiar with the technique. So this was Evan McCasco from Steve McCarroll's lab in 2015 kind of came out with this idea that being able to, in a high throughput way with a microfluidics device, pair up cells and beads, you would be able to do high throughput sequencing in a very rapid manner. And the idea is that these beads have polynucleotides on them that can capture the mRNA, and you can process everything in a high throughput manner. So again, the flow device kind of looks like this. In the lab, we can make these. So we used ones that are not commercially prepared that we make ourselves. Here is, a model of the flow showing a bead coming along here and a cell coming, right through here. And of course, this is highly slowed down, but this happens, thousands of times a second. And again, for the people that aren't familiar with this technique, really the beads are the key to the process. So what they ended up doing was basically doing this pooled synthesis method where they can take beads, they pool them, split them into A, G, C, and T synthesis rounds, pool them back together, and repeat this 12 times. And that's how you can basically get the same sequence on all the beads for the PCR handle, but then switch to having a very specific cell barcode where every oligo on the bead has the same exact sequence, and then use, again, random sequencing or synthesis for the UMIs and the polyCs. So you can then take this information, and once you sequence it, for every given mRNA, you get your cell barcode that lets you map it back to the cell, and a UMI for PCR correction and things like that. Again, once you get this information, you can recover this information by your 2 reads. So you do paired-end sequencing, but the first read basically just gives you your cell barcode and your UMI, and the second read gives you your gene identity and lets you actually record your mRNAs. So finally, we get to, what am I actually going to do with Drop-Seq? So anecdotally, I actually, wasn't really sold on single-cell sequencing kind of at first when it first came out, because, again, I'm pretty old school, so I was around back when we were first doing RNA-seq, and we're talking like 50 million reads per sample and stuff like that, and then these Drop-Seq single-cell papers come out, and you're like, oh, you're getting 50,000 reads per cell, how are you getting information from that? But luckily, my old PI kind of convinced me during one meeting with him that Drop-Seq was the way to go, and so we've been doing this for about a year and a half now, getting Drop-Seq. And so the first thing that we really wanted to do was just look in a kind of unbiased way. Let's take a bunch of breast cancer cell lines, let's do Drop-Seq on them, and see what we can pull out. And so we took a variety of different breast cancer lines, and if you remember, there's all these different molecular subtypes of breast cancer, so we spread our cell lines out across basal, Claudin low, the HER2, and the luminal lines. And we did that data. So I'm going to most of the analysis that I will show you is from Seurat. I'm sure that everyone is pretty familiar with it. It's pretty much right now the kind of the default Drop-Seq analysis pipeline. I've played around with a lot of other ones, but I tend to stick with Seurat for the most part. So our overall analysis pipeline, I use the McCaskill Java programs for all the read alignment, use STAR, all their cleanup steps. Everything is very standard there. And then I pull it into Seurat, do the standard, some more standard quality control and filtering. And overall, we do the principal component analysis, TSNE, for the reduction in the dimensionality. Then I cluster cells with the traditional Seurat clustering and look at gene expression differences. So when we first did all 10 cell lines and did the TSNE clustering, what we really hoped to find, so our main goal, going back to what I described previously, is that we expect across the different molecular subtypes to have kind of commonalities between these stem cells. So different stem cell populations might be similar across the different cell lines. So what we were really hoping for, I don't think we realistically expected that this would instantly pop out, but in a best case scenario, that you'd be able to take the stem-like cells that we know are present in all these different cell lines and see all of those grouping together and maybe have a couple of their separate populations of these non-cancer stem cells and basically recapitulate that one figure where you had across different molecular subtypes, different cell populations. But of course, basically what we really got was, and I'll show this in more detail, but each of the cell lines sits very independently, and either by, almost in any clustering method you use, Seurat tells you that they are all very distinct and doesn't really mix up the different cell types. And so before I get into that data, I'm not going to show any of the data today, but we've actually been working with Jun Lee and Simon and other people from the Midas group in looking at further and better ways of analyzing this type of data, but that's still work in progress right now. The idea would be to try to pull out more subtle differences based on transcription factor networks and things like that.
Do any of those look like cell cycle state?
I will show you a nice graph that shows you that, yes, but cell cycle is obviously pretty much the first principal component that pops out, and it's one of the only ones, but neither of these kind of completely split into like a cell cycle. If your question was, is one of these groups cell cycling and the other non? No.
No, that wasn't it.
Okay,
It was
but
more
yeah.
like G1S.
Yeah, in
different
each
phases.
cluster, it very well defines like cycling versus non-cycling. Yeah. So we first started looking at our cell cycle markers, or our stem cell markers. And so, again, we had ALDH expression, and there's two different like isoforms of ALDH that really drive things, and that's ALDH 1A1 and ALDH 1A3. But this is a good example of why we know that these cell lines didn't really like pull all the stem cells together, because for the most part, only one of our cell lines, SUM159, has very strong expression of ALDH 1A1. And so it sits here off to the side. When we, for example, looked at ALDH 1A3, we saw that our two cell lines that were reasonably strong in expression sit here with, again, a spattering of other cells that are ALDH 1A3 in the rest of the population. If you remember, CD44 was another one of the markers and has reasonable expression across. And finally, one of the other, both a molecular subtype marker and a stem cell marker is HER2. And so we saw that very strongly expressed in the two HER2 positive cell lines. And it is interesting that you saw a good amount of HER2 expression in most of the other cell lines. And again, this is an interesting, it's anecdotal, but basically it matches with something that Max and other clinicians had found, was that HER2 lines have went from being one of the most poor prognoses to one of the best prognoses because we developed a lot of targeted therapies to HER2. But it turns out that if you are misdiagnosed as HER2 positive, given HER2 treatment, but then turned out that you weren't really probably like a full HER2 amplified tumor, that you do much better. And so basically, that it's probably been found that all of these cells in all of the tumors are probably an important stem cell marker. And so treating almost any patient with Herceptin can actually have positive benefits, which makes sense given that we see HER2 positive cells throughout all of the tumors.
The HER2 agreed with the cell of origin. So ALDH also agreed with on the epithelial side, and the other one CD24 on the Claudine low side.
Yes, so that really agrees. For the most part, CD44 matches up with what you would think. And that like the more mesenchymal basal triple negatives have higher expression of CD44, and the luminal ones here have lower expression of CD44. And the inverse is true for CD24.
So what you really want to find the cells in the middle, the stem cells.
Yeah.
Either on this arm or the other arm. Do you have a cell line that has a character can find a system in another 1,000 cells?
Yeah, so that's basically where I'm going to get to, is that we realized that, even with this broad data, we really we couldn't focus in on those. So eventually what we do is we don't have a cell line that has like a pure positive population, but we did Drop-Seq on sorted populations. So we sorted out the ALDH positive cells and do Drop-Seq specifically on those. Yeah. And so, and this is another reinforcement that kind of, yeah, everything makes sense in this data in a broad sense. Our epithelial lines have high EpCAM and ESR. Our mesenchymal lines have very high vimentin. But as you can see, the reason I'm showing you a lot of these patterns is basically what June mentioned, is that the reason that we obviously don't get all of our stem cells together is there's just so many genes that are just wildly different between the two cell lines. So even stem cells in all these different populations are still, the ESNE is going to be driven by vimentin and EpCAM being strikingly different between all of the cell lines. And that's what's really driving it. And then so I'm showing you all the genes that kind of drive it.
Sorry, this plot is based on all the genes, this ESNE plot, or just based on the stemness, the genes that are markers for stemness?
It's basically, I think this is based on the top like 500 most variable genes, but not specific to stemness or anything other that we knew a priori from breast cancer.
Sounds like you're catching the wrong end of the train. If those markers, if you assume those cell lines, they're all having some sort of like features in common, then those genes presumably they won't differ much. They want to be different from one cell line to another. So maybe you should look at the ones that are, I don't know, the ones don't vary too much. Do they show up as clinically known stem cell markers?
Yeah, so that's a good point. I mean, so we think that, yeah, it's a hard problem, right? So not only do we think that all the stem cells might have the same signature, but they're all such a very small percentage of any given tumor. Like only 5% of these cells would be stem cell markers. So we haven't done what you were suggesting, which I think would be a good idea to eliminate some of these genes and look at them more.
My understanding from this plot is that you are actually catching the genes that are characteristic of different cell lines.
Yeah.
They are different between cell lines, so they end up in different positions on the ESNE plot.
Yeah. Yeah, so, and that's one way, but how do you decide, like, which of the, I guess you could just keep eliminating all the most variable genes and kind of only do your ESNE on, genes that are more in common. I mean, what our hypothesis was, and what we worked with June and another computational postdoc and some other things, Sahar, was to still look at all of the genes, but look at the transcription factor pathways. And those might be more kind of in that realm where they wouldn't be so drastically different across the cell lines that maybe we'd be able to see that.
Right. So that's actually a good idea. Look at the functions that are robustly shared among the different stem cells. So maybe it's something like a Notch.
Yeah. So yeah, and so when you, for example, if you pull out Notch or things like that, they're really lowly expressed for the most part. But yeah, you'll see like a handful of cells in each group. But yeah, I guess what we really need to do is kind of isolate those and then do maybe the clustering just on those.
Okay, so this is going to sound like a very unpopular opinion, but I've had my experience with unsupervised methods, and frankly, I feel like they're kind of like a Rorschach test, where you just keep trying until you get something that looks good. And how do you really judge whether one method is better? Like whether you use all the genes, the top 500 most expressed genes, top 100 differentially expressed genes, like, or the most variable genes, like, is one really any better in the end? I feel like all you're trying to do in the end is get a pretty picture.
Yeah. No, I would be pretty much in that camp. But yeah, I think a lot of this Drop-Seq analysis and single cell analysis in general kind of fits that mold where like there isn't a consensus way of doing all these analyses, so you can have this bad tendency to like just keep doing the analyses until you get the answer you want. In particular for clustering, I think that's one reason that we haven't played around too much with like which genes we picked. We just kind of used the topmost genes, and we know that this made sense in this way, but yeah, we probably could have played around with it. But the idea is, yeah.
So basically, you can't really say for sure which method is better for sure.
I mean, I think this gives the true. I believe in this result in that I know that these cell lines are so different from each other that I'm not surprised at all that they sit apart.
What can you do with this plot besides just looking at a picture and saying, oh, we can separate all 10 cell lines from each other.
Yeah.
We already knew ahead of time that they're not the same cell line.
Yeah. No, I mean, and we did it with the hope that maybe it wouldn't be this, but as June mentioned, I think the follow experiments is what we did, was take individual cell lines and do directed sequencing on, our stem-like populations to look more specifically at those.
Yeah,
Because from this.
Because I was also thinking, maybe you could use this plot to find, well, you already have an idea of a few marker genes a priori, right? I mean, you could probably do some sort of, I don't know, independent component analysis, something like that, to find even more marker genes that haven't really been noted in the literature. I mean, if you're already doing T-linear clustering based on every single gene in an unbiased fashion, maybe there's some other marker genes that people haven't really noted before, and you could just visualize that just to confirm.
Yeah, and I will get to that once I do the ALDH sequencing, that's definitely one of our goals. So in the interest of time, I'll skip over these, because again, most of this is just the same thing. But I did want to show, back to your question, that you will see that in general, for any given gene that I showed up here, the population of that cell line is basically yes or no most of the time, and very homogeneous across the cell line for that. There are some differences like in BT20 here, but for the most part, that's how it works. But the cell cycle was, of course, probably the biggest differentiator, where we know that in principal component 1, in driving a lot of this clustering, that whether a cell was cycling or not was really defined that. But that's not surprising, and these days we regress most of that out, and most of the data I will show you from now on actually is regressed out. But I did have this original picture where we weren't regressing out the cell cycle quite as much.
Here, you within each block, you score cells for the cell cycle activity. So those with lesser colors are more likely to be the quiescent or dividing further kind of cells in the dish.
Mm-hmm.
But if you also score the stem cell character using the Malta's machine learning,
yeah,
would you at least see the slower dividing ones tend to have a higher stemness score within that block?
Yeah, that would be really interesting. So I haven't used that machine learning paper as much as I should to basically take their block of genes and cluster on it and figure out whether that's affecting those populations or not. I think we're thinking about doing that, Simon and myself as well. So to get around this issue, so I think everyone is basically in agreement with what we said from this data is we didn't find all these very specific stem-like populations in an unbiased fashion across all those different cell lines. And so what we wanted to do was then go more specifically and do Drop-Seq just on the cancer stem cell populations. Now, the main downside to this is that now we're relying that the markers that we know about, which is basically CD44 plus, the combination of CD44 plus and 24 minus, for ALDH, that those are correct, and we know they work, but they might not be the only marker, they might not be the best marker, but now we've kind of biased ourselves based on that information. So I did it from 2 populations. So the first one that I want to just mention was SUM149, so that was one of the cell lines that I just showed. But we used a reporter system to mark the cancer stem cells. Now, it's still basically the same markers. We used ALDH 1A3, driving an M-cherry, and then because it's a reporter system and it's really tricky to do like CD44 plus 24 minus in a reporter system, you'd have to have more reporters, and we are working on that. But we used a general mesenchymal reporter from Vermitten driving GFP. And so from these cells, we would basically have 4 populations. So we would have the double positive cells. So those should be the cancer stem cells that kind of have dual properties. So they sit in the middle, they have ALDH, and they're a little bit mesenchymal. The double negative, we wouldn't really know exactly what populations those were, so we would, in general, expect to kind of just ignore the double negatives. Then we would have our really mesenchymal cells on one end, and our really epithelial cells on the other end. And so we would be looking for a lot of stem-like properties in our double positives. Then finally, we also took the Vario 68 cell line, and we just sorted for ALDH and CD44 plus 24 minus. Though, in the end, this population was so small, it was like 0.001%. It was probably basically background. And that's another caveat to this, is that not all cell lines will have all the different markers. So I will only talk about the ALDH positive, because essentially it seems like CD44 plus 24 minus isn't a good marker combination for the Vario 68 cells. And then we use bulk as a control, which basically is the absence of either of the markers. So when we took the Sum 149 reporter after all of our Drop-Seq, and in general, we were going for about 1,000 cells of each, and there's about 600 or 700 cells of each after filtering. Now, this looks like a big jumble, and somewhat we weren't super surprised. Again, this has, again, just a cell line that we have sorted, but in general, all the cells still come from that same cell line. But what I will show you here is, and sorry, the dots are a little clearer on my screen, but basically, the GFP-only cells and the M-cherry-only cells really sit distinctly in their separate quadrants of this giant ESNE blob. Now, the interesting part is, and we still haven't figured this out, and it's certainly a big caveat, is that the double positive and the double negative kind of sit on top of each other. We suspect, and I'll show you some data that reinforces that the sorting worked and everything, with the normal variability of single cell data worked pretty well. But we suspect that maybe the double negatives ended up as artifacts that have lost the reporter for some reason, and so they're technical negatives, and they probably should have been in the other populations, but we're still not 100% sure about that. But to verify that our sorting, worked, we can look at, for example, ALDH1A3, and that was our reporter for ALDH1A3 drives M-cherry, and so you can see that you definitely get the strongest signal from the M-cherry as well as the double positive. But and something that's not surprising, but probably not appreciated as much, that there is still a lot of purity issues when you just fact sort something. So there's plenty of ALDH1A3 expression in the double negatives in the GFP only, but there's a clear enrichment that's very statistically significant for the double positive and the M-cherry only. If we look at M-cherry or the Vimintin, we see again something pretty similar. The GFP only and the double positive are definitely the GFP only has a significant enrichment. There seems to be a little bit of M-cherry only Vimintin signal, but compared to the double negative, it looks pretty good. So we are pretty confident that we are getting some information from the reporter and can separate out these populations.
These black dots don't match with the one, the leftmost one. That's black dots.
Yeah, I think that helps to. We did notice that. I mean, obviously, it means that there's still there must be an extraordinary number of zeros here and some high. So it's basically bimodal compared to a little bit less of a bimodal distribution here. And that might back that the double negative is truly you have some true double negatives and some contamination from what should have been an M-cherry positive cell or something like that. Because yeah, it looks like it should be pulling the violin plot pretty high, right? But there must just be so many all negatives or a few positives. Was our assessment. So if you take these cells now and ask Seurat to do some unbiased clustering, you end up, this is just a small outlier population of dead cells that we would normally pull out and redo the analysis. But you can see that you get basically, 5 true or 6 real clusters. And so we looked across those clusters to look at the population, the percentages of the cells. And so this also backs that, again, particularly the GFP only and the M-cherry only populations are very significantly different. But while the double negative and the double positive still are sitting kind of on top of each other. And so look at this graph. So this is cluster 0, cluster 1, all the way up to cluster 7. And this is in the numerical form. So this essentially is the same data from here, just, I guess, flipped upside down. So the M-cherry only has the highest enrichment for the cluster 0, which have also the highest enrichment for the ALDH 1A3 expression. So this is basically the ALDH 1A3 cluster. And you can see the other cell types there.
Sorry, so the 6 clusters, you define them? Or it's discovered in unsupervised way?
Unsupervised clustering by by Siraha.
For all 4 samples?
Yes, exactly. So you can basically ask of those populations, because again
do you pull them together?
Yes.
Combine as like one data set and ask for how many clusters are there?
Correct.
I see.
Yep. Yeah. So all of this data was the combined data sets put together. I've analyzed them independently as well, but we found that it was kind of most interesting to again put them all back together and ask that can you pull out those stem-like populations when they're all in one large group. So we were interested in, as I mentioned, that we think that cluster 0 is the M-cherry only / the ALDH cluster, and ALDH does come up in that cluster. So we did some gene set enrichment analysis, and we do get a lot of the stuff that we would expect: stem-like targets early up, gastric cancer early up, thyroid up. But nothing that really stood out. We still don't get a lot of SOX2, OCT4, NANOG, some very pluripotent stem-like genes that we know are important in cancer stem cells. So I wanted to, again, we had this data, and we looked at the Vario68 sorted ALDH cancer
What
cells.
kind of expression level do those genes have?
Which
The ones you wanted to see, but didn't.
Yeah, I mean, very rare,
Yeah, so aren't you in the territory where with single drop sequencing, you're going to be just stochastic as far as seeing things and not?
Yeah, so that's why we tend to, still do, high populations of cells, like 1,000 cells, and what we've been looking for is pushing that further. But I think, there's the work that we've been doing is, again, with Jun Lee, and is the idea that those stem cell transcription factors, SOX2, OCT4, and NANOG, for example, very lowly expressed, but their pathways downstream lead to genes that are higher expressed. So we've been trying to look for the downstream targets and more networked analysis and things like that, but that's still kind of ongoing.
Do they have, or you estimates of efficiency of recovery of transcripts?
So basically, they say, the quoted in the paper and seems reasonable based, on some back of the envelope math, is about 10% of the mRNA of any given cell is capturable. So yeah,
Yeah, there's a lot of stuff that's present at only a few transcripts per cell.
Yeah.
Or even less. They're only part of the time.
Yeah. So that, the nice thing is, though, that at least there's no evidence against this, that 10% that's captured should be across, whether you're the highest expressed gene or the lowest expressed gene, your chance should be approximately equal to get captured. And so if you can look at enough cells, you can still hopefully, still see your occasional SOX2, OCT4, and NANOG and be able to discriminate those populations. So that's what we've been hoping on.
If you already know their populations.
Well, or if, if they're truly in the ALDH, have only SOX2, OCT4, and NANOG, so you can see that they still only come up in those ALDH populations. But I completely agree that those particular genes are still very rare. So nicely, the Vario68 data looked even better than our reporter, because we're just staining for this cancer stem cell marker. It's a little bit more of a snapshot in time. So when we still put them both together and clustered, we see a significant shift in the two populations. And when we plot the ALDH either on that or the violin plot, we can see a significant enrichment in that population. And so when you ask Seurat in an unbiased fashion to cluster those, you get kind of two separate populations with the cluster number 2 is interesting, but for the most part, it's driven by this stem-like signature between 0 and 1. And again, this is similar to before, where you can see the ALDH expression. And the cluster number 1, this is 0, 1, is the one that's specific to the very 068 ALDH. So when we pulled out those genes and did gene expression analysis, we do get ALDH 1A3 and a lot of other interesting genes. So this does make a lot of sense and gives our best data. And back, to the question before, this is where we're hoping to find like the new markers of ALDH in stem-like cells. Is any of these genes, and so we're looking at finding which of these genes might be extracellular and being able to sort on them and see if those also are markers of cancer stem cells in breast cancer. So those questions were mostly looking at ALDH itself compared to the bulk. So we also wanted to kind of dig down into the ALDH cells themselves, because for the most part, you could argue that I could have done almost all this, in a bulk sequencing method if we're just going to sort out ALDH positive versus bulk. And so when you take just the ALDH positive cells themselves from the very 068 and cluster them, you end up with getting some unique populations. And we're still in the process of digging through these, but one of them, certainly looks pretty interesting. Though it might be the cell cycling. It's obviously there's certainly some cell proliferation stuff here, but it's probably mixed in with some interesting biology because not all of these are cell cycling, and this data was regressed on cell cycle analysis. So if it's still bleeding through a little bit, there's probably some other interesting biology in there. So again, we'll use these markers to kind of see whether ALDH itself can be further refined by different markers. So our future directions, so, continuing some of these advanced transcription factor analyses with June and other collaborators, I think will be really key because, again, like we keep finding, for the most part in a lot of these analyses, stuff we already knew. We're confirming that ALDH is there and that the mesenchymal versus epithelial spectrum is still there. But we're really not pulling out the biology behind the cancer stem cells and understanding how those are functioning. So that's one of the things that we're really interested in. I'm sure everyone's really well aware, I focused on Seurat here. I continue in myself try all these new data analysis pipelines and packages that come out. I think that, kind of back to your comment, that even across different pipelines, I usually redo the analyses and, they usually look basically the same, but you never know if they look different, like which of those was the right way, and you just keep going until you get one that gives you the result that you like. I'm wary of, that kind of bias that can be introduced by just trying a bunch of different stuff. So we're going to validate the sorted cancer stem cell Drop-Seq by using those gene lists to hopefully find some new cancer stem cell markers and being able to test them in vivo in mice and understand whether hopefully we are getting some new biology. And then we're also working in collaboration with Indika Rajakoski to do some single cell Hi-C. Maybe there was a new paper that came out looking at from UW, a guy I can't remember right now, and I should, but yeah, Shinduri's lab, doing the RNA-seq and attack-seq on the same cells, so maybe we can do that from cancer stem cells and do that. So with that, I'd like to thank my mentor Max. We have a lot of great collaborators and people in the Wisha lab, and I have grant support from the BCRF and a postdoc fellowship from Eli Lilly. And so with that, I will take any other questions.
For Drop-Seq, is there a significant batch effect?
Yeah, so there definitely is. So even particularly from any two samples that are the same cells sorted by different times, I see drastically different levels. And the main thing that I see is kind of that background ribosomal and mitochondrial percentage that a lot of people use as kind of a metric for how healthy your cells were. But I see pretty big differences in that set of genes. But it seems to be able to be regressed out, in Syrah pretty well. But there's definitely some reasonable batch effects there.
But to answer that question, you can benchmark it by out of the 10 cell lines, run one of them second time.
Yeah.
Throw it in to see if it's a new island on its own or not.
Yeah, and so I've done that. And so basically, yes, it would, if you don't do very much regression and you plot them, they'll be their own island. But if you regress out the mitochondrial and ribosomal and cell cycle, they tend to basically be on top of each other. And still not 100% though. So yeah, basically there are definitely some batch effects.
When you sort the double negative ones, is your predicted stem cell enriched population.
So in that, for the reporter system, you mean?
Your flow sorting the full population.
Yeah.
The double positive ones, your hypothesized stem cell population.
Yes.
Can you just go in and see if it has more stem cell signatures than the other single positive populations?
Yeah, and so at least, so we did that. So basically we tried to look for, so the problem is that we used one of our main markers, ALDH, as the sorting, and it basically looks about the same as the M-cherry only, right?
But the gene-efficient data, use the gene-efficient data to score the stemness.
Yeah, so we haven't done that with some third or a second measure of stemness, like using the machine learning, gene set as the scoring.
All the genes that you have not chosen for sorting are available for you.
Yeah. So we looked at, for example, SOX2 and OCT4 and NANOG, and they're just too lowly expressed and don't seem to be different. And we looked at, CD44 plus looks about the same. But otherwise, using a less biased, that machine learning paper, I think would be the way to do it.
And your proliferation marker should be only in the ALDH positive fraction.
Yeah. So again, with cancer, based on Max's paper, we kind of treat the stem cells as quiescent compared to the rest, but it's still pretty relative in that they still have pretty high proliferations.
I think this is a slow chase for something that's elusive. And stem cells are minorities in a dish that are hunkered up, they don't replicate, they don't breathe oxygen. Get addicted to glucose. Can you just throw a lot of bad stuff killing the stem cell, or just enrich it with a sledgehammer and throw the Yamanaka factor on it?
Yeah.
Get 1,000 cells there to be a positive control. Right now, we don't know whether they are below the threshold of detection. We don't have a positive control. Those are the stem cells.
Yeah. Yeah, we've thought about that. I mean, and I think we're getting to that point where we're going to have to. I mean, obviously, the downsides are that no matter what you do, you might change. You're going to be affecting the stem cells in some way, and you don't know how much you're really pushing them and how artificial. Particularly if you try to throw there are induced cancer stem cell models that are the Yamanaka factors put onto a cancer background, but I've never been super convinced that they're not making some weird new crazy cell that doesn't represent true cancer stem cells. But I think we're getting there that we would need to do that.
I just worry about the data we currently have. The variations are driven by batch effect, by cell cycle. The true stemness is a shadow of a shadow that's very, very minor.
Yeah. Yeah, it's true.
Are these all low passage cultures?
They are all relatively in the Wisha lab, they're less than 30 passages from ATCC, but of course that doesn't
That's a lot
really
of
exist. I mean, yeah, it doesn't.
Like, if there's stem cells that aren't highly proliferating, you'd think you'd be diluting them out, and you'd be dependent on things, I guess, that transform back to that state.
Yeah,
It wouldn't take long to do that. Not many.
No. We basically know that of the, so I take that back. So 9 of them are like super high passage, like again, like, you know, well above, those effects would become significant. Besides the Vario-6/8s that are a low passage cell line we derived ourselves in the lab. But we do know that of all those cell lines, they do have still cancer stem cells in that you can take the ALDH positive versus the ALDH negative, put them in mice, and get increased tumorigenicity. So there's a...
They have to be
They do
switching
have
back,
a...
you'd think.
Yeah, there's a homeostatic balance between like the differentiation and maintenance of the cancer stem cells over long periods of passages. But in general, I didn't show the data today, but we do have in vivo PDX, patient-derived xenograft data that is lower passage, always grown in mice. But similarly, we still have a hard time picking out the cancer stem cells in that data as well. And so, again, it might go back to whether it's a threshold issue.
What do you have to do to derive the line from, like, patient sample? Is there any...
A little bit of trial and error, but for the most part, it's taking a chunk of the tumor straight from the OR and putting it in the mammary gland of a mouse and letting it grow. And then, you know, maybe 50% of those grow, 50% don't. And the ones that do, you just, once that mouse, the tumor gets big enough, you chop it up and put it into more mice. And you basically just expand it forever in mice and never let it touch
But
plastic.
these lines are actually tissue culture lines.
Yes, these lines are different than the patient-derived xenograft lines.
So what do you have to do with the, you know, because are you selecting for kind of rare things and not looking at typical cancer?
Yes, I mean, absolutely. So you'll notice that all these cell lines, again, are very homogeneous, and it's because they basically had, like, you can take a chunk of tumor, throw it in a dish, and 3 cells will grow out of that, out of the million that you threw in the dish. And if you redid that 100 times, you'd get 100 different cell lines. But the only issue is, the only thing that makes it okay at least a little bit is that, you know, the SUM159s that we use are somewhat similar to the SUM159s that the different lab uses. So at least what we call SUM159 is kind of standardized, even if it in no way represents what that original tumor really was.
Alright, so
I think the first thing first is that, you know, for your data, your single cell data, what is the average? What's the norm? Is it also correlating very well with bulk cell RNA-seq data to start with?
Yeah.
That will be the figure number 1. To see, you know, your experiment works, so what's fine. And then go from there to identify stemness-like things.
Yeah, we haven't done the full RNA-seq on all those cell lines, but I've done
I think it's
qRT-PCR.
public. You can just go public the data.
Yeah, that's true. Yeah, most of those will probably have public RNA-seq.
Alright, thank you.
Thank you. Yeah, there's so much to do.